Started here. I'm just going to share my screen. Just so attendees, so you are aware, this is a webinar, and so your video and microphone is disabled. If you would like to share your questions, we'll be taking them at the end if there's time. Hopefully there will be. You can just share those into the chat, and I'll be relaying them on to the right PI at the end. So, all right, let's get going. All right, welcome to day 3 of the MAISRC virtual showcase. My name is Meg Duhr. I'm on the MAISRC staff as our research outreach specialist. I'm really excited to be here introducing 4 of our new MAISRC funded research projects and their principal investigators. This announcement is the culmination of a 3-year process in which MAISRC consults with the research community, management professionals, and the general public to identify knowledge gaps and set research priorities. From there, the center develops a request for proposals based on those highest priorities, and we then evaluate and select proposals for funding. It is an intensive, prolonged, and highly collaborative process. It's also a highly competitive process, and the strength of these proposals, as well as the critical AIS research needs that they are meeting, reflect the significance of this work. These 4 projects in particular are an excellent reflection of how our process should work and how an established research center like MAISRC can drive long-term research advances, getting us closer to the ultimate goal of effectively managing and even someday eradicating AIS. You may have attended past showcases or heard about various developments in genetic methods, like mapping the zebra mussel genome, or advances in environmental DNA methods for detecting early invasions or cryptic species. Or perhaps you just learned about projects to shape a statewide AIS surveillance program. All of the projects you're about to see build off previous work and take us to the next level, addressing the methodological questions, public awareness needs, and partnerships needed to actually implement this work. Our researchers are going to be presenting lightning talks, and during this session, they will introduce you to their projects. Time will be tight, but if you have a question, post it to the chat. We'll try to get to it at the end. So with that, I would like to introduce our researchers. Dr. Daryl Gohl leads the University of Minnesota's Genomics Center's Innovation Lab and is a research assistant professor here at the U. Dr. Gohl's work focuses on developing new techniques for genomics-based measurements and genetic manipulation of complex biological systems. Dr. Gohl was part of the team that sequenced the zebra mussel genome and is now working to use this genomic information to develop methods to genetically manipulate zebra mussels and to identify genetic weak points that could aid biocontrol efforts. Dr. David Fulton is an assistant unit leader in the U.S. Geological Survey's Minnesota Cooperative Fish and Wildlife Research Unit and adjunct professor in the Department of Fisheries, Wildlife, and Conservation Biology here at the University of Minnesota Twin Cities. He is a national expert on the human dimensions of wildlife and conservation social science, serving as a principal investigator on several dozen applied social science and interdisciplinary research projects to better understand human values, attitudes, and behaviors concerning wildlife and fisheries management. Many of his research projects are conducted in close collaboration with local, state, and federal fish and wildlife management agencies to address key issues confronting these agencies. Josh Stumke is a senior research scientist at the Natural Resources Research Institute at the University of Minnesota Duluth. His research focuses on fisheries and aquatic ecology within lakes and streams of the Great Lakes Basin and inland Minnesota. Josh's recent research interests include researching how native species interact with invasive species and identifying mechanisms of overland spread of aquatic invasive species. Collectively, this work will help researchers, anglers, and recreational boaters limit their risk of transporting invasive species as undetected hitchhikers. Lastly, we have Dr. Amy Kinsley. Amy is an assistant professor in the Department of Veterinary Population Medicine at the University of Minnesota Twin Cities. Her research focuses on using advanced epidemiological modeling techniques to develop tools that support decision-making surrounding management of invasive species. infectious diseases, and aquatic pollutants. Most recently, she has been working on a legislative report with MAISRC on the design of a statewide surveillance and early detection program for Minnesota. Right. Okay, so I am going to turn it over to Dr. Daryl Gold.
All right, thank you, Meg. So as Meg mentioned, I lead the innovation lab in the University of Minnesota Genomics Center. And I'm going to tell you today about a project that we're working on together with Dr. Mike McCartney, a zebra mussel expert, and Scott Valentine, a professor at the University of Wisconsin River Falls, where we're working to apply a technique called RNA interference to try to identify candidate genes that could be high priority targets for potential biocontrol efforts in the future. Together with Dr. McCartney, I was part of the team that sequenced the zebra mussel genome over the past several years. This was a large group effort. It involved collaborations between a number of different groups at the University of Minnesota, as well as a number of external collaborators. And we released the sequence of the genome last summer. And although this headline caused some consternation among some members of the research team, this is kind of the goal of sequencing the genome, or the rationale for sequencing the zebra mussel genome, was to take that next step and to try to identify targets for manipulation and identify genetic weak points or genetic Achilles heel of zebra mussels. So this project that we're working on now represents, the attempt to take that next step. So what we have with the zebra mussel genome is really a catalog of the genes and the building blocks of the zebra mussel. But one of the challenges that we have is that this is a very large catalog. So the zebra mussel genome comprises about 1.8 billion letters. And so for some context, Tolstoy's War and Peace is about 3 million letters long. So this is equivalent of about 600 copies of War and Peace, which if you were to stack those up, would be roughly the height of an 11-story building. So this is a lot of information and a lot to sort through. We do have some, a bit of a head start on kind of digesting this information. So in the context of the genome project, we identified a number of genes that are involved in critical processes, such as shell formation, the formation of the byssal threads or attachment fibers, as well as genes that are required to respond to very, very warm temperatures or thermal stress. So based on both that information and the fact that genes that are involved in critical processes, like critical biological processes, tend to have a lot of evolutionary conservation. So we can go and use information about gene function from humans or from fruit flies or from other organisms to identify genes that we think, based on just their basic cell biological functions in zebra mussels, are likely to have relatively severe effects if we were to manipulate them. So based on both this sort of like knowledge base from other organisms and also these experiments that we did in the context of the zebra mussel genome project, we've identified a list of about 150 genes that we view as really high priority targets and potential candidates that when disrupted could cause potentially severe fitness defects in zebra mussels. So the next challenge that we have in attempting to manipulate these genes is that while zebra mussels are this huge problem and infestation of lakes in Minnesota and around the world, one of the ironies is that you can't actually grow these for long periods of time in the lab. So we can take them into the lab and cultivate them for a period of time, but you can't robustly kind of propagate them generation after generation. And so some of the techniques like applying CRISPR or applying gene drives or some of the things that other speakers in the symposium have discussed aren't really an option for zebra mussels, at least not at this point, until you know those tools and technologies to be able to robustly culture them are actually developed. So to solve this, or you know, instead we've turned to this technique called RNA interference, and this is a process that was discovered in the worm C. elegans, and essentially one of the advantages of RNA interference is that in worms and a number of other organisms, there can be constructs that can induce this RNA interference process can be fed to organisms. And in the case of C. elegans, it's common to feed them bacteria that express these double-stranded RNA containing constructs. And what the effect of this RNA interference is that this is a photo of a worm that's expressing a molecule called GFP, or green fluorescent protein. And when worms are fed bacteria that express double-stranded RNA against this GFP, what you can see is a lot of this green signal goes away. And so basically the expression of this gene is knocked down. And RNAi has been shown to work in a wide variety of different organisms. There's just a few examples here, you know, everything from fruit flies to humans. And importantly, it's been demonstrated to be effective in other bivalves as well. So in oysters, for instance, RNAi has been used to induce phenotypes. And RNAi is the sort of cell biological origin of RNAi, or the reason that exists is, you know, it's triggered by a number of different types of double-stranded RNA. So one form is double-stranded RNA that comes from viruses. So, you know, this is thought to be, at least in some part, a kind of an ancient antiviral defense mechanism that the cell triggers when it sees foreign these double-stranded RNA molecules, which tend to be of foreign origin. And so what we can do is use this process that occurs within cells to trick zebra mussel into basically mounting this sort of antiviral response, or this gene silencing response against its own genes. And so what we plan to do is use this list of, or use this collection of genes that we've identified, potential biocontrol targets that we've identified, and to screen them using RNAi. So we'll express these constructs in bacteria that will feed to the zebra mussels. And then as filter feeders, they should take the bacteria up, and then the RNAi can have a chance to, you know, act on the zebra mussel genes and silence them. And then we plan to screen all of these targets through a collection of phenotypic assays that the team has been developing. So this is an example of one such assay. So in this case, what we're doing is feeding the zebra mussels bacteria, E. coli bacteria, that are expressing this green fluorescent protein. So you can see that, you know, when this bacteria is added to the media, these test tubes are glowing green. And then if we feed this to live zebra mussels over a period of a couple of days, they will filter this bacteria out of the water, and this GFP signal goes away. So this is really a readout of viability and sort of metabolic activity of zebra mussels. And what you can see is that in these control mussels that have been azide killed, or killed by adding in sodium azide, the GFP signal persists. So we'll be using an assay like this one, and then also additional assays that we're developing to measure the attachment of the byssal thread attachment fibers, to measure shell formation, and also to measure fertility. And we'll be screening through a large number of candidate genes to try to identify genes that have significant effects in these assays. So with that, I'd just like to thank all of my colleagues in the University of Minnesota Genomics Center, all of our collaborators on the Zebra Mussel Genome Project, and especially Mike McCartney and Scott Ballantyne, my collaborators on this project.
David, that's really exciting. Or Daryl, sorry. Thank you, Daryl. I'm gonna turn it over to David now.
Okay, thanks very much, Meg, and thanks, Daryl, as well. Let me get my presentation up real quick. Okay, and hopefully you're seeing a screenshot of the presentation. I'll be following up a little bit, and this is great juxtaposition of these two talks, because I'll be speaking about genetic biocontrol of invasive species, but I'll be focusing on understanding attitudes and risk perceptions among the public of these technologies. And just to provide some background, and Daryl just did a great job of doing that for me, but over the last several years, what we've seen is tremendous increase in new synthetic biology, genetic engineering techniques, with a lot of promise for very different conservation management actions, including the control of invasive species. Dr. Gohl has just given a great example with zebra mussel here at University of Minnesota. Another example here at Minnesota is Mike Smansky's lab and their development of engineered genetic incompatibility with zebrafishes with extension onto common carp that has real promise as a potential biocontrol of common carp. As we've seen these techniques developed, what we're facing now, or what we're experiencing, is growing calls not only among the researchers developing these techniques, but among many conservationists to explore the benefits and the use of genetic biocontrol technologies. And as these technologies mature and move towards that ability to implement them, at least technically implement them, there's feeling that there's need to understand how the public perceives the benefits and the risk of using genetic biocontrol technologies to help guide policy and policy dialogue, to develop management strategies about if and how implementation will occur. And as we're on the cutting edge here in Minnesota, both with perhaps control of carp and zebra mussel using these techniques, it was thought that this would be a good time to examine what public perceptions are. So there's a broad conservation context to this issue, and the project that I'll be conducting has 4 key objectives. One is to understand the attitudes, risk, perceptions, and level of support for using these genetic techniques to control, in these 2 examples in Minnesota, common carp and zebra mussel in Minnesota. These are 2 widespread, commonly known invasive species among the general public and among stakeholders. We're also, as Dr. Gohl just demonstrated, we're at the cusp of being able to use genetic biocontrol techniques for these species. So we thought they would be great case studies here in Minnesota. We also want to understand the antecedents and consequences of level of support for using genetic biocontrol techniques in these 2 specific cases. What we mean by this is what actually predicts level of support among the general public or stakeholders for these biocontrol techniques. And then what does level of support mean for management actions and strategies? If they're technically feasible, but we find that the public has heightened risk perceptions of these techniques, what does that mean for management? What does it mean for implementation? How do we move from there? A third objective is to understand the general preference for using genetic biocontrol techniques beyond these 2 specific case studies. That is, what attributes of the management environment or management policy drives preferences for different genetic biocontrol techniques? When are they generally acceptable? When aren't they? A final objective is to explore and gain an initial understanding of potential concerns of tribal communities in Minnesota with genetic biocontrol techniques. We want to make sure that we understand and engage the tribal communities so that we can understand any unique concerns and issues they might have with these techniques. The study itself will be conducted in a broader theoretical context. In the conservation social sciences, there's a well-developed body of social science theory about risk perceptions and about invasive species. We'll use this established literature to develop established psychometric approaches for understanding values and ideology, attitudes and risk perceptions, both to genetic biocontrol techniques, but also to the invasive species themselves, the common carp and zebra mussel. What are the attitudes? What are risk perceptions of these invasive species? How does that play back to the perceptions of risk and acceptability of the biocontrol techniques? And finally, we'll extend some of the work that McDonald and her colleagues recently conducted in New Zealand by using discrete choice experiments to understand the preferences of using genetic biocontrol techniques more generally. That is, what attributes of the management strategies themselves drive acceptance or preference for certain policies or strategies of using genetic biocontrol techniques. The methods that we'll be using will be collecting data via social survey with the public and key stakeholders in Minnesota, a final target sample size of 3,200 completed surveys, 1,200 among the general public, 1,200 among lakeshore homeowners, since they live in the context of where these invasive species occur, as well as 400 anglers and 400 boaters. We'll focus on online data collection. We're focusing on online data collection versus traditional mail surveys because it's going to enhance our ability to present information to potential respondents to help them understand these genetic biocontrol techniques. Dr. Gold just presented things fairly simply, and as we can all see, this is fairly complex stuff. So to send out a basic mail survey and ask people what they think about it, the responses aren't probably going to be very accurate. We're probably not going to get accurate attitudes. So what we need to do is present information to them about the complexity of these techniques and hope that we can gather risk perceptions and attitudes information that is fairly well informed. Also, over the last 20 years, what we've seen is an erosion of response rates in traditional mail surveys. This past year, with our experience with COVID-19, those response rates to mail surveys seem to erode even further. And so we seem to be at a crossroads where using the technology of the web might actually enhance response rates over mail surveys. With our tribal communities, we'll focus on conducting interviews and focus groups, both with tribal biologists and community members, again, to get those perceptions of unique concerns that these tribal communities might have with these technologies. The outcomes of this project, what we anticipate is not only developing upstream information about public risk perceptions concerning genetic biocontrol, but we want to use this upstream information to provide a context for a future policy dialogue here in Minnesota concerning the use of genetic biocontrol. As Dr. Gold just described, we're very close to having these biocontrol techniques ready and available. So the next step is to develop the policies and strategies as we move towards implementation that the public is going to embrace and accept. And so that's it.
Thank you. Also very exciting and critical work that needs to happen alongside with the advancement of these genetic tools. So yeah, we look forward to hearing more about this in the future. Next, Josh, you're up.
Okay. So hopefully my screen is visible now. All right. I'll be talking about the project starting up, optimizing eDNA, monitoring for multiple AIS. I'm the project manager and Dr. Gretchen Hansen is co-PI with me on this. And we're going to be helped immensely by Dr. Chanlan Chun here at NRI and also Dr. Eric Larson over at University of Illinois. So I'll kind of set the stage on what we're actually doing. So aquatic invasive species are continuing to spread among Minnesota water bodies. If you look at the infested waters list that's kept up by Minnesota DNR, where they track about 17 species, if you average the new detections annually over the last 5 years, there's about 53 new AIS detections every year. And that isn't new lakes. That could be any, either a new lake or a new species found in a lake that already had another species in it. So on average, about 50 detections are being added to that list a year. And we know that AIS early detection is critical to facilitate a rapid response for control and preventing spread to other nearby lakes. And I guess control, if it's possible or applicable, that particular species. But the basic point is we need to know where AIS are in order to slow their spread. However, early detection using traditional methods requires substantial investment, where investment is the time of personnel and the expertise that these people need to have. And this limits the number of lakes that get surveyed. I have done traditional surveys for, I believe now about 20 years, and you get very good at it, you get very efficient at it, but you still are limited by the amount of time you have and how many places you can visit. And there's also the added challenge that physical detections may still lag years behind AIS establishment because the populations may be low for a long time, and it may take a lot of effort to detect them with traditional methods. So this means that there's missed opportunities to prevent spread. So I have a little image at the bottom where somebody's leaving a lake and there's no signage, so there must not be any AIS here, off to the next lake. And it's just this concept that if you think that you're partaking in an unrisky behavior, you may not be as vigilant at checking your boat or where you go next or how frequently you visit different lakes. So in reality, maybe nobody has ever looked in this lake for invasive species, or if they have, those invasive species simply haven't been detected yet. So it's this false sense of security that may be happening out there in the landscape. So what about eDNA? Environmental DNA, or eDNA, is DNA released by organisms in their environment, like skin cells, mucus, feces, and gametes. Collecting water is one way to get eDNA. eDNA has the potential to detect AIS invasions early without the high cost of traditional sampling, but those cost savings can only be realized if eDNA methods are optimized for multiple AIS. So the same amount of effort could lead to detections of multiple species, and if users have confidence in the results. So the problem is eDNA results vary among users because methods and sampling strategy vary. And this isn't unexpected. eDNA is a relatively new tool, so there's a lot of work happening all at the same time. And so there's a lot of different approaches to using eDNA as a tool. In our project, we'll optimize methods for eDNA detection among 4 invasive species which impact Minnesota waters. So we're going to work with common carp, zebra mussel, rusty crayfish, and spiny water flea. And we have 2 project questions. The first is, how does lake character and seasonality affect detection probabilities of multiple AIS using eDNA? So to answer this question, we're going to visit 21 lakes 5 different times between April and October. The lakes will vary in size, trophic status, and region of the state. And during each sampling event, we'll measure water quality, collect 250 milliliters of surface water from 10 different locations, filter that water through a 1 micron cellulose nitrate filter, and then extract and quantify target DNA copy number. From this, we expect to learn what types of lakes are most appropriate for eDNA as a detection tool, and whether our target AIS detections are greater during lake turnover or at times of increased biological activity. And examples of those are in the spring, carp spawn. Are we more likely to detect them when they're biologically active? And the same goes for zebra mussels that release veligers, typically in July, that are free floating stage. In sometime in midsummer, rusty crayfish will molt, where they shed their exoskeleton and build a new carapace that hardens over hours to days. And spiny water flea abundances increase as you approach late summer and early fall. So as the populations of spiny water flea continue to build, are you more likely to get positive detections using eDNA if you sample later in the year? So that's the aspect of seasonality. What is the effect of the timing of your sampling? For our second question, we wanted to know what are the optimal combinations of eDNA field and lab methods. And here we're looking for sensitivity. What are the most sensitive methods? We'll pair 5 lakes. We'll sample 5 lakes once with many paired test groups. We're going to look at filter materials, comparing cellulose nitrate filters with a novel sorbent developed by Dr. Meg Duhr. Filter material pore sizes of 1 versus 5 microns. How much water is filtered, 250 mils versus 1 liter. Where the water is collected from, nearshore surface water versus offshore surface water or offshore water collected near the lake bottom. And quantification method, qPCR versus droplet digital PCR. ddPCR is a newer quantification method. It's been around for about 10 years, but it is much more sensitive than qPCR. And so we wanted to test whether or not it may be an appropriate strategy to use during early invasion when DNA concentration numbers of your target organism are expected to be very low. And we're going to look at some test groups of preservation and extraction methods. So our baseline is ethanol preservation with Kygen extraction, and we're going to compare that with the kit that comes with the sorbent versus a CTAB preservation and chloroform extraction method. So we're going to look at different choices you can make in the use of eDNA methodology and see which of these seem to be important in making getting positive detections when you have concentrations of DNA or organisms that are low. For this activity, we're also going to pair traditional survey methods to determine AIS presence where water was collected. And we think this will be important because it will help us explain DNA yield and any unexpected positive or negative eDNA results. From this, we expect to learn which methods are most sensitive for detecting our target AIS and therefore would be best suited for early detection of new invasions. So we're really trying to focus in on the sensitivity of your choices and methods. Our goal for this project is to create method recommendations for the field applications of eDNA when monitoring AIS in Minnesota. So managers, AIS specialists, and non-government organizations have guidance and confidence when to use eDNA as a monitoring strategy. We intend to disseminate our findings through peer review publication, presentations at conferences and MAISRC events, and creating an eDNA best practices guide, which will describe our findings and recommendations in an easy to understand format. And I have a little tongue-in-cheek picture of what we're going to have as an output. And it's going to be likely an electronic document and hopefully not so big that nobody wants to start reading it. But we do hope to make these recommendations in a way that can give guidance to folks that actually want to start collecting water in lakes of interest. So with that, that's the end. My contact information is up there. And I also thank MAISRC for selecting this project for funding. So thanks for tuning in.
Thank you, Josh. Yes, this is going to be a great project and very timely in light of other surveillance work happening at the Genomics Center and the scaling up of this need for large-scale surveillance. So thank you for sharing. And Amy Kinsley is up next.
Great, thanks, Meg. So today I'm going to share with you guys a project that I'm working on with my co-PI, Robert Haight from the USDA Forest Service, where we'll be working to improve the efficiency of watercraft inspections through coordination and cooperation. And so watercraft inspections are a key component of prevention, providing a lot of benefits. They help prevent the spread or the introductions to uninfested water bodies. They serve as a form of education to boaters on the dangers of AIS and then how to keep their boat clean so that they're not introducing infestations to uninfested water bodies as well. And they can be used to collect information about boater movements. And so despite these benefits, there's a lot of difficulties in managing watercraft inspection program. And some of those difficulties are just the sheer number of boats that move across the landscape, and also the large number of water bodies that we have here in Minnesota. In addition to that, we know that there's limited resources. We can't have inspectors at every single water access point. They can't be there all of the time. And so that leaves program managers grappling with decisions regarding their strategies of where to place those inspectors and when. And some of the things they may be grappling with are whether they prioritize inspectors at infested water bodies or uninfested water bodies, and they may have different reasons for that, why they would choose one over the other. And so currently we've been working to develop an optimization model that helps decision makers or program managers make those types of decisions. And so the current model helps select lakes for inspection stations with the goal of maximizing the number of boats that are moving from those infested to uninfested water bodies. And so that takes into account boat movements into a county, within a county, and then from what's inside that county out to other water bodies outside of the county. And currently considers 4 different invasive species. It looks at zebra mussels, starry stonewort, Eurasian water milfoil, and spiny water flea. And we've done that through a combination of techniques. The first was using linear integer programming. And so that approach works to find an optimal value, which could either be used to minimize or maximize something. And so for this, we used it to maximize the number of inspected number of inspected boats. And so it's either minimizing or maximizing an objective function, and that objective function has variables that represent different actions, so inspection or not. And then that inspired us to create a heuristic network-based approach that we could use to feed into a user interface, so that different county managers creating strategies could access that information in those model outputs. And so that heuristic approach used structured techniques called social network modeling, or social network modeling based techniques that can analyze interconnected components. And like I said, we use that to build a user interface. And I shared a little bit about that at my talk on the first day that we're calling AIS Explorer. And I'll touch upon that a little bit further in the talk. And the current model is informed using boater movement data. And that data was derived from the Watercraft inspection program surveys. And so we've used data that was collected from 2014 to 2018. And part of the Watercraft inspection program survey included questions about where boaters were coming from, what that previous lake was that they had visited, and where they intend to visit next. And then to overcome biases inherent to those survey methods, and then also to account for water bodies that were not inspected. That data was statistically analyzed using some machine learning techniques called extreme gradient boost analysis. And that then gave us estimates that were aggregated at the annual level from lakes in the state, even if, like I said, even if they were not observed in the Watercraft inspection program directly, so that we could help people make those types of decisions about where to place the inspectors. And so overall, we have data about movement between over 9,000 lakes. And so the current model outputs look like this. And so I'm showing here just a mock-up of the AIS Explorer app. And so this is not the current model outputs, but gives you an idea of what those outputs look like. And so the user can select the county of interest and what their management goal would be. So what proportion of boats they would want to inspect. And so here we have it just set at about 90%. And the user can go in and select the different species of interest, and then they can get the data out as a CSV, or they can get charts or maps as well. And so I'm not showing the map here, but the map just shows the locations that were selected for inspection. And so the output then also provides a table with the water bodies that are ranked, with the highest ranking lake, so lake number 1, would be the lake that, if you placed an inspector there, would intercept the largest number of those risky boats that are moving from the infested to uninfested water bodies. And the output also includes the infestation status and the DOW number for those lakes. And it also says the total number that fell within that proportion of boats that was set for the management goal. And in addition to the list, we also have a chart that gives planners an idea of how many inspection stations they could add before they start to lose out on the value of adding new inspectors. So like a curve of diminishing returns in a way. Where on the x-axis, we have the number of inspection stations that are added, and on the y-axis, we're showing here the proportion of boats inspected. And the users can toggle over to then see the map, which, as I mentioned before, would just show those locations that were selected. The app also allows for an option where the user can customize the included lakes, and the default would be the lakes that came up on the list of rankings, and they can either include or exclude the lake of interest so that they could tailor the output to their needs, because potentially some lakes come up on the list that actually aren't options for them to consider, and or they have different strategies that they want to consider other than just the optimal. So we wanted to allow for that as well. So in our new project, we want to expand on these existing efforts. And here, the overarching goal for this project is to quantify the benefits of state level coordination and between county cooperation and watercraft inspection and plans. And really, we want to explore different questions regarding scale, so about planning at the state level versus the county level, and then what the benefits of coordination are. And we want to foster collaboration between counties as well. And so two of the main aims of the project are to estimate the benefits and costs of allocating watercraft inspectors for the whole state. And so that would be our example of coordination. And we also want to look at fostering county level information and resource sharing. And we want to do that through developing collaboration networks. And when we say networks, we mean these social networks using the voter movement data. And so that would be a form of collaboration. And so for the statewide plan, we're going to implement the integer programming model that we used in the project that we are currently working on for the entire state. And we want to look at the optimal statewide scheme, the different optimal county schemes, and then look at varying degrees of coordination between the two. And we want to estimate the gains and losses between, or the gains and costs between those varying degrees. And we expect to have a proportion of risky boats that are inspected and what the different financial resources are needed to carry out the scenarios. And then for the county collaboration networks, we want to apply the heuristic network approach that we're currently using in the project we're working on for each county. And we want to identify which counties contribute to the in and out of county movements in that optimal strategy. And so we're calling these the cooperation networks. And here I'm showing an example of what a cooperation network might look like. And this is using preliminary data. So this is just for conceptual purposes, but I'm showing the top 10% of between county movements for Otter County. And so we can see here, Otter County is the county of interest, and we can see that it's connected directionally. So if the arrow is going outward towards Douglas County, this is suggesting that Otter Tail County boats are moving into Douglas County. And so these lines are also weighted so that those lines that are thicker are representative of larger numbers of boats. And we're hoping that these collaboration networks can open up discussions for potential cooperation where people can compare watercraft inspection plans because we know that right now they're only putting inspectors within their county. But if they could coordinate with those outside the county and have an idea of who those ideal coordinators are, that could be more helpful. So we hope to include that in the dashboard that we have created that we are planning to release within the next month. And so we want that to be part of the AIS Explorer. I showed a YouTube video of the promo for the AIS Explorer as a YouTube link. And for those of you who weren't able to make it to my talk and are intrigued, you can jot down the address here and get access to the link, and you guys can take a look at that as well. We'll be sending out emails to let you guys know when that is ready and up and running. And we're just working on some quality control right now. So with that, I want to thank Epi Interactive, who's the group that helped us form the dashboard. I want to thank all the collaborators that helped us with the Voter Movement Network and optimization project, including those county managers who gave us feedback and have helped us to date. So with that, I will turn it back over to Meg and open it up, hopefully for questions.
Okay. Thank you so much, Amy. Hey, if you wouldn't mind, would you put that YouTube link into the chat box so everybody has it there?
Great idea, absolutely.
Thank you. Cool. Well, I hope for our audience here that it's becoming clear why we decided to lump these 4 projects together. There are some great collaborations that are possible that are already happening here at MAISRC. I especially love that we're actually taking these emerging eDNA tools to actually develop protocols for how people can actually use them, which is very critically needed. And then using the work of Dr. Kinsley and these modeling systems that will help us prioritize which lakes to do additional surveillance. Hopefully that can get us to a point where we are actually doing large scale, maybe even statewide surveillance, which is what is needed at this point. I might kick it over to Daryl. Would you comment just quickly on some of the capacity that the Genomics Center brings? And we do understand that all resources have been focused on the COVID response, justifiably, but maybe you could tell us a little bit about how the buildup of your staffing and lab resources might be used for AIS surveillance in the future?
Yeah, and I mean, I think the University of Minnesota Genomics Center has been one of the sites doing COVID testing in the state, and it's amazed me the parallels kind of between that work and the types of surveillance, but both doing the detection, kind of like diagnostic, which would be similar to like eDNA, but then also using, genome sequencing and using that genetic information to understand the spread of the virus. There are a lot of parallels with what we want to do to detect and monitor invasive species as well. And yeah, so we've also been working on some eDNA assays for zebra mussel that are based on the genome. So I think that there are a lot of opportunities to work with Josh and his project and help scale those measurements. I think the work that he's doing is really fantastic, and attacking this in a really systematic and thoughtful way.
Yeah, and there's a lot of interest from lake associations and other volunteer groups in embracing these new methods. And so once we can get protocols and timelines established, I think people are going to be really excited to take it to the next level. We do have one question so far. So folks, if you do want to ask questions, we have a few minutes. One question came in for you, Amy. The writer is wondering if there are serious advantages in your project goals with incorporating technology like the eyelids. And for those who might not know, I can't think of the acronym right now, but eyelids are autonomous systems that are set up at boat launches that aren't staffed with a human boat inspector, and they're motion activated, they're solar powered, and they will play a recording with a reminder of AIS prevention methods. And a lot of them also do have like a camera. So the idea is that it could be used in a law enforcement situation. If somebody pulled out their boat, obviously infested, or launched a boat, the camera would pick up vehicle identification information, in theory.
Yeah, I think that's a really interesting question. And I think that would apply to the voter movement networks and validating some of those estimates, especially for answering that question about those points where we don't have watercraft inspectors. And I think we've thought a lot about how we would go about that. And this point comes up from time to time. And I think we also come up with the same issue, which is that potentially these devices aren't distributed evenly enough or used often enough to really help us validate that. But we also talk about using other types of approaches to go through that. And one part of this project that I didn't touch upon, and one of the aims, is doing a sensitivity analysis. And so going through the data and trying to figure out what parameters in the model are influencing our model outputs. And so I think within that, we can begin to adjust some of these networks and really try to understand how sensitive our model outputs are to our estimates. And I think that then could lead to maybe further questions like this, like implementing new technologies to try to dive in there. I think at this point, though, for this specific project, including that is just a really interesting concept to consider. Yeah, thank you.
Okay, and we did get one more question also for you, Amy. They're wondering if your modeling effort is, or this project as proposed, is looking at the current county collaborations that are happening right now. And then will you be including any recommendations on citing mobile decon units, or is it just inspectors?
Right now, we're just looking at inspectors. And then going back to looking at current collaborations. So we've worked directly with some county managers to get feedback on the current models that we've developed. And so we'll continue to seek out advice from county managers. And I think one thing that would be really interesting is to understand further about how those collaborations have developed and how they've been used. And so if anybody is interested in having those conversations, I urge them to reach out to me, and I see that I can insert my email address here. So I'll go ahead and implement that, or go ahead and insert that. Yeah, and so I think that's something that we're interested in including for sure. So we are, we have that as a source of input for the data as well when we're looking at the cost of resources. So if anyone's also interested in collaborating on that component, we would be happy to chat with you as well to get an understanding of what strategies you're using and how much manpower, how many hours you're placing inspectors out there, how much those costs are. All of those types of details are appreciated for sure.
Cool. And then we got another question about does the model, or is the plan to look at collaboration between the DNR and county agencies? Or I'm sorry, county managers?
Yeah, so when we're looking at the different levels of coordination between the state level and the county level, I think we'll address on points, those types of points. But right now we're looking at conceptually that optimal state level model and what that outcome would look like, and then those optimal county level models. So I think there's implications for that, but that is not necessarily specifically the question that we're asking, because my understanding is that the DNR strategy is likely to be different than what that optimal strategy is, considering a variety of different species and the boater movement network that we've created. So there might be similarities and there might be parallels, but not explicitly.
Yep. Okay. Great. Well, that is all the time we have. I need to tee up the next webinar here. Thank you so much, attendees, for coming and learning about the new talks. Next year at this time, whether we're in person or virtual, who knows, but we'll have more updates from the team here after they're half a year or so into the work. So thank you, panelists. That was great. And we are so thrilled about these projects and look forward to hearing more. Thanks, everybody. See you at the next one.